And then we would hope to, open this to the public, and people could walk through and, collect chestnuts or whatever. And we would hope to allow school groups in, and we have some little trails planned around. So this is kind of the next step that we're thinking that we would get these started really before landscape scale restoration would happen, so we would have the long-term data. Now I'm going to get into a little bit more of the regulatory stuff, and it's not very exciting to talk about, but it is important, and it would be the next step for, well, current step for us and next step for you guys, I guess. And starting with planting permits. So any transgenic plant, before it can be planted outside, in order for it to be planted outside, needs to be under a permit. And these are certified specific locations where they can be planted, need to fulfill certain obligations for fencing or control, or they can't be on sites that are known to have endangered species, and they need to have limited access, and just a number of things. And then to maintain the permits, there's a lot of paperwork, and you need to report each time a new plant goes in the ground and submit another report, and you do annual reports. You need to keep maps of exactly what's planted where, maintain the identity of each tree, keep everything labeled, whether or not it's transgenic, and allow USDA inspectors to come in and see basically when they choose. Usually it's about once a year. So it's not a trivial thing to maintain permits just for testing. So we have 7 locations around New York that are permitted test sites for us. Some have 3 trees and kind of demonstration plots. Some have many hundreds of trees for kind of more research plots. But this would probably start with a single site for that's maintained. Among other things, so just recently, a big step for us is that USDA has allowed, on certain sites, under certain conditions, allowed us to potentially do some open pollinations. And that is, in order to grow some big trees, we need to let them produce pollen. And until 2016, we had to cut off every catkin before it ripened. And you can imagine for a tree that gets much taller than you, that would be nearly impossible. And so we actually were coppicing trees only to prevent flowering, to maintain our permits. And so it's tough to do research that way. So allowing open pollination was a big one. Controlled pollination is another kind of level where if you don't want to remove the catkin every single flower, you can put a bag around it, and then you can collect the pollen, and under certain conditions you can do pollinations that way. But one of the conditions that we've mentioned already is maintaining this 400-meter or about quarter-mile zone around every transgenic planting, where you have to certify there are no compatible relatives, nothing that could be pollinated. If by chance one of your pollination bags broke and pollen got out or something like that, you need to be able to show the USDA, well, they're not going to walk around with you, but you need to certify that there are no compatible species that could be pollinated by chestnuts within 400 meters of your test sites. And then that's just one of many regulations that go with this. So permits for test plantings are one big thing. Those max out at something like 10 acres. It gets a lot more complicated if you want to do more than 10 acres of test plantings. But that's kind of, in order to do your testing, you need to plant them outside, and permits from the USDA are the way to do that. Thinking beyond permits, before we can actually distribute these, use them for restoration, we need to get approval from 3 different U.S. federal agencies. FDA, because Food and Drug Administration, because chestnuts are food, eaten by people and animals. EPA considers the gene we're putting in a pesticide. Even though it doesn't kill the fungus, we argued that it's not a pesticide because it doesn't kill the fungus. EPA's lawyers disagreed because of how the laws are written. USDA actually evaluates a couple of different components, but generally I'd say kind of environmental safety and so on. So there are 3 different petitions that we're currently working on, and we're getting ready to submit these to all 3 agencies for them to review, so we can hopefully start to distribute these. In addition to the official regulatory agencies, there are a few other groups like the Forest Service, National Park Service, that are not regulatory. They don't have to sign off on something or approve something. but the actual regulatory agencies will be consulting with them. So it's important that we are in contact with them, we're showing them what we're doing, we're not trying to hide anything, you know, we're trying to reach out to these groups and make sure they're absolutely clear on what we're doing, and if we can get their support, that would be a big help, even though they're not actually regulatory agencies. And then big environmental groups, things like Nature Conservancy or National Wildlife Federation, high-profile groups that most people are familiar with. Again, they don't have to sign off on these, but the government groups are going to be paying attention to what those big groups are thinking. So the more support we can have broadly from the public, from the big groups, is very beneficial. I will move on for the sake of time. There's a lot of detail here. Kind of general regulatory considerations. Everyone asks, how long is this going to take? And for each agency, it's a little bit different, but really generally, really preliminarily, let's say about 2 to 5 years after we hand in the submission. And if everything goes really well, I guess hopefully 3, but it's really hard to tell. And they actually, most of the groups have timelines where after they receive this, they have 22 months to give an answer of such and such. And in reality, it rarely works that way because it's the government. And they can delay a lot of things. But, yeah, getting back to that one later. And there are costs. And in some cases, like submitting a petition to the EPA starts off with a $335,000 fee. But there are groups, land-grant universities are one group. There's a group that's part of the USDA that's exempt from that fee, from that, it's called a PREA fee. So we're working with that USDA group. They are going to hand in our petition and save us $335,000. So there are exemptions from that, and so it is prudent to pay attention to things like that. And another point I'm going to make here is that once registration or non-regulated status is granted, once we have approval to plant and distribute these things, it's for what's called a transformation event, or a line basically that we make of American chestnut that has the transgene. That covers all offspring of that American chestnut. So we could breed it, we could, you know, and that's really our goal, is that we would breed these things with the wild surviving American chestnuts, and that provides some genetic diversity for the start of a restoration program and prevents this, you know, genetically uniform forestry scenario that we want to avoid, and we want some more genetic diversity with these plantings. However, this covering all offspring would potentially include Chinquapins. So I don't want to push you in this direction, but I want you to be aware that if we get approval for transgenic American chestnuts, one possibility would be breeding them with Ozark Chinquapins, and then potentially back-crossing with more Chinquapins to keep those Chinquapin traits. And that would avoid the entire regulatory process. There would be no regulatory oversight. If we have approval for the chestnuts, and if the gene is put in by breeding, no restrictions. Again, totally up to your group. I'm not trying to push you. That's just something to be aware of. Other considerations. We're always trying to kind of maintain this balance between privacy and security on the one hand. We don't want extremist environmental groups to come and rip out our plots because they're scared of GMOs. On the other hand, we want to be transparent. We don't want to be hiding anything and covering things up and being secretive. We are frequently giving talks and tours and inviting people to check out our plots, and really community involvement, I say here, and buy-in are key to success. Without engagement from whatever interested community is going to be planting these trees, we won't be successful. So maintaining, yes, balancing both of these is a challenge. You guys already, sounds like, are dealing with some vandalism on plots and whatever, but that's just something to keep in mind moving forward. We have fences primarily for deer, to keep the deer out of our plots, but we lock everything to keep casual intruders out also. Also, just generally, both tree research and any government regulatory submissions are going to take a lot of time, so that combination might take even more time. So this is a matter of patience, and I'm hoping that my kids and grandkids can see mature American chestnuts. But we need to start somewhere for this to happen. So, a little bit of what we've seen so far as feedback. Every time, I can say without exception, that we've talked to the government regulators, USDA, EPA, FDA, all three, even Canadian regulatory agencies, because American chestnut range extends into Canada. Everyone we've talked to, the government, the scientists who are going to be reviewing our petitions, they have all been really optimistic and helpful. They can't bend the laws. Their job is to uphold the laws, but they have all been very supportive. They want to see our petitions. They're encouraging. Our questions are more at the kind of higher political levels than at the scientific levels, but that is what we've seen so far. We've seen a lot of support, especially from the New York chapter of the American Chestnut Foundation, but also in recent years the national group has really been supportive. As far as general public perception and feedback, overall it's been very positive. There have been a few people who are kind of opposed to this idea of genetic engineering, but compared to, say, agricultural biotechnology and even people who are opposed to a field of genetically engineered corn, see work with the American chestnut. Oh, that's for restoration, this environmental purpose. Okay, there are people who support our work even if they oppose the same technologies in agriculture. So that's been encouraging for us. We get a lot of presentations, and the vast majority of people we talk to, the first response we get is, how soon can I plant a tree in my yard? I want a blight-resistant American chestnut tree. So that is, that's exciting also. Thinking about kind of regulatory stuff a little more broadly, there are lots of agricultural crops that have gone through the regulatory process. Most of the corn and soy and canola and cotton that's planted in the US is genetically engineered, and the big companies that make these have whole staffs of literally hundreds of people to do what my boss and I are doing of this regulatory process. But there have only been a few trees that have gone through the regulatory process and been approved for planting. And those are all orchard or nursery type trees. Papaya, apple, plum, eucalyptus is kind of a maybe right now, orange is in process. And so those are basically profitable trees. No one has done this for any forest type tree. Chestnut will be the first there. No one has really done this for any nonprofit, for good reason, because it's an expensive process, and so the ones that have gone through are profitable. And this technology basically hasn't been used for restoration yet. I would argue that there's so much potential there, it's a matter of time until other groups are doing this too, but chestnut's the first. And there is research into other forest-type trees. We've done a little bit with elm, other groups are working a little bit with ash, walnut, some of the western pines are threatened by pine beetle. Hemlock is another common one that a couple labs are really doing preliminary work with hemlock. Lots of threats to trees, as I'm sure you're well aware. And some of those groups have, at least with walnut, I think, they have some planting permits. They're doing some test plantings with transgenic trees. But no one else has submitted petitions yet for release. And another interesting thing is that even though we haven't even submitted petitions for broader planting and distribution yet, chestnut is, to some extent, being seen as a success story. We've actually been contacted by a group in Hawaii who's dealing with a tree called the ohia tree. And it might sound familiar, but this ohia tree is very important historically and culturally and environmentally. There are these unique birds that live on the ohia tree, and just in the last 5 or 6 years, this tree has been threatened by an invasive fungus. And they're seeing significant dieback in some places. They don't even understand how the fungus spreads from tree to tree yet. But last year they contacted our research group, and I actually got to go to Hawaii and talk to this group about ohia tree preservation, restoration. They're not ready to jump on board with the transgenic ohia tree, but they want to hear about our work with chestnut, and what types of things should they be thinking about as they start any sort of restoration or conservation effort. Even coral reefs. You're probably aware that ocean acidification, ocean temperature increase are threatening corals, and there's bleaching and coral diebacks. So, not surprisingly, there are coral research groups looking at coral conservation and breeding. They invited someone else from our research group to come and talk about chestnut research using biotechnology to preserve a threatened species, as that might be applied to coral. So really interesting and really exciting, getting some attention from other conservation groups. With that, I'll wrap up. Happy to take any questions, and I think that I probably used more than my share of time, but appreciate your attention and happy to help however I can. Yeah.
So, if we were to do this for Ozark Chinquapin, there's still a couple things we have to figure out, and one of those things would be like the growth auger. The growth auger and the nutrients that need to be present in that.
It would probably be a matter of starting with the chestnut one and doing some tweaks to figure out how does Chinquapin grow best in culture. Yeah.
Could you talk about that briefly?
Yeah, sure. So we have one line, I think, of Ozark Chinquapin embryos that came from another lab in Georgia, so I don't have any idea about their origin. They might not be good trees. They're probably not what we'd want to start with for any sort of...
We've never confirmed anything from Georgia.
Right, so we don't know what they are.
It's possible, but there's some from Virginia.
Okay.
It's possible, and I'm speaking without looking at it. I'm just telling you what we know.
Yeah. So anyway, we would want to get from you guys good quality embryos that were from known pure trees. Okay, yeah, so as far as next steps, kind of tweaking stuff for Chinquapin, we've done a similar thing with Chinese chestnut. So we had our tissue culture procedures pretty well worked out for American chestnut. We knew how they grow and what different types of media needed to be present for each developmental stage and so on. And then when we started putting Chinese chestnut into culture, they didn't grow the same way. Yeah, they're closely related, but they didn't respond the same way to the growth media, for example. So we needed to, for each of those stages, go through and, okay, what concentration of, I don't know, sugars or growth hormones or whatever are optimal for Chinese chestnut, and just optimize that process. So it would probably be similar. And it took a little while, but it's not that complicated. It takes time. Is the main thing.
Not 10 years.
A person. No. More, a year or so. And it depends, whether someone's already working on it. That would probably be, a master's scale project to figure the whole thing out for Chinquapin.
I've got a question for you. So I work for the U.S. Army Corps of Engineers, and for us, if we were interested in trying to get a test plot permit, would we still have to go through the whole process as well, even being a government entity?
I think so.
Yeah.
I'm not positive about that, but I think that's unless you work for this branch of the USDA, APHIS it's called, I don't think the requirements are any different.
Back to your tadpole study.
Yeah.
Even though there was a very trivial difference between the transgenic and the hybrid and so in the standard one, but there was a difference. What on earth could it be that the tadpole was missing? Do you have a speculation? Can you speculate?
I don't have any idea as far as why a transgenic leaf might cause a difference.
I was kind of surprised there's even a difference.
Me too. And it could be that, we looked at 30 tadpoles, and if we looked at 100 tadpoles, we might not see a difference. It might just be a sample size. And it's hard to say whether there's something missing from the transgenic leaf that the tadpole needed, or something present from the transgene that was causing a difference. The scale of differences that we saw would have a lot more data than what I showed. And most of what we saw is not statistically significant. So that's what, take into account the sample size and is this difference real? If we repeated it, would we see the same difference? That's what some of the statistical tests are telling us, that, it's not a significant difference. So yeah, we would want to repeat that but other things like, size, the mass at the metamorphosis, there weren't differences there. So, it's trying to take all of those different things into account. And are you looking at size or survival or development at 3 weeks or development at 8 weeks, or, you know, it's, some of them showed differences both ways, that, you know, okay, maybe a transgene helping, maybe it's not. So then on the whole, I don't know how to balance that.
That sounds,
But
it sounds like a
it's-
very true,
we're paying
small,
attention to it, right.
yeah, very small.
Right.
So that, the regulation process only happens for a transgenic seed, right? So what about if you just wanted to plant just American pure chestnuts? Can you do that without
Sure.
going through the process?
Oh, absolutely, yeah, only because it's transgenic,
Mhm.
yeah.
And where-
So we can distribute.
What's the best place to get those? Like, I'd be very interested in doing test plots, or my next question is it even worth the effort to do ones that aren't blight resistant?
Yeah.
I mean, is it worth
Yeah, that's
spending
a
that time? I don't know.
very common question. Why should I plant American chestnuts right now if they're going to die from blight? And for our project, where there's basically no natural resistance in American chestnuts, what we are hoping is that in a few years, if everything goes well with the regulatory process, we'll be able to distribute some blight-resistant transgenic American chestnuts. But like I said, we want some more genetic diversity. We want variability there. And so we are strongly encouraging people to plant non-transgenic chestnuts. One, so they get used to it. They know how to plant them, they know how to take care of them. But also that if things go well and we're allowed to distribute transgenic ones, plant them nearby, allow them to pollinate, and there's some more diversity. Rather than distributing the ones that we've produced with a minimal level of diversity, allow people to outcross them, or you might even be able to distribute pollen. So here's a little slide with some pollen on it that we've collected. You pollinate your tree that's in your yard and collect the nuts, send it back to us for testing if you want, plant them if you want, and kind of carry on. That's how we would envision next generations of this
So
procedure.
is there like a wait list for every student for you, or?
We
'Cause I just-
always say,
We've got, you know, at the core, we've got quite a bit of land that
Yeah.
I could do test plots on for that.
Yeah, no, we
Doing
could-
Chinquapin test plots, I would like to do American chestnut test plots.
yeah, we could
as
send
well.
you
Well,
some American chestnuts.
Okay.
I have some little brochures up here, but I also have a card, so send me an email, and
Okay, I'll grab one.
I don't know if I can send you hundreds, but I could
No, that's
send you
fine.
dozens. And it would be next fall when we collect them.
Right, obviously.